I know.
Okay.
Okay, boys and girls, we're live with rational empiricist, who I shall call Andrew, and Speed of Sound, who you can call Mike or whatever the fuck you want. It doesn't matter to us. And we're going over a video from, let's see, January 1st, 2021. That was debunking Gutsick Gibbon. Junk DNA. We're going to talk about junk DNA. Finally,
that was kind of the first 2 hours of it.
Yeah.
I mean, we could go over it if you want, but I don't feel like you want to go over 2 hours of them just blathering about how junk DNA isn't junk.
Yeah, I do want to go over some of the things that they said as a. I think I can't remember I can't remember who I agreed to do this with, but there are two we're gonna have a talk about junk DNA and DNA structure.
Okay.
And I kind of want to go over it with not without getting 8 people in the room, just a few of us, and go over some of the fine points of what this junk DNA thing is all about, really. I mean,
yeah, that would make sense.
Today we're talking about this video, Scott. We are going to try and talk about a video.
Is Scott Duke there?
Yeah. Yep. He's on chat.
Like, who Scott are you talking to?
Oh, amazing. What are we talking about today? Yeah, an hour after the weather report, so I'm burnt out already. Okay, so should we play it?
Sure, yeah. Quick question. Dr. Card, before you go.
Yeah, I saw that. You wanted to ask. Go ahead.
So if I believe in evolution, personally. So I want to, I guess, ask a challenge question, to see kind of the terms creationists sometimes use, and I think I've used this example with you standing and you Nephilim free, but I just want to ask you, Dr. Carter, about it. So to give a specific example of a mutation, there is a gene in Drosophila called Scoot that controls the expression of bristles across the adult body of the fruit fly. And a mutation in a specific, I don't know which enhancer specifically it is, but this enhancer is pleiotropic. And this mutation that changed a T to a G in the species D. santamea, in comparison to other species of Drosophila, like when you're comparing the DNA differences, basically you find this mutation. This enhancer mutation caused a decrease in the number of bristles near the genitalia of the fruit fly, and in male flies caused a increase in the number of bristles along the forelegs, which are used to for mating success, I guess. And so I guess just before I get to my main question, so you would say that mutations tend to destroy information, correct?
They can destroy, but they can alter. There's very, very few examples of where, how do you improve on the handiwork of God, is my perspective. Some people will claim being able to have ice cream as an adult is a beneficial mutation. I would disagree with that, but it's, if you're starving to death and the only thing to eat is goat's milk or cow's milk, there's obviously an advantage to that. But it's when you're trying to say that all mutations are negative or near neutral, there's obviously benefits if the environment becomes extreme enough. Like the people that have sickle cell anemia, or one of the most common mutations is where you have trouble regenerating enough NADPH, so you don't make enough glutathione, which is one of your
body. Can I say something?
Yep.
So isn't it? It's like it frustrates me that they like they use the word beneficial and deleterious almost synonymously with their usage of information, basically. Yeah. So is it related to fitness? Is it related to just the DNA in general? Have you seen this before?
Yeah, they have they're very unclear about information, and in the last week or two, a couple of them have tipped their hand out a little bit, and they think information is something that creates like an arm, and that arm is a positive benefit to the organism, and it's only information if it does that, right?
Right.
So these bristles now, I'm not sure if you ever explained it that clearly to them, but these are on two different species of Drosophila, right? So
Yeah, it's on like the bristles in general are on Drosophila species, but like this mutation changed the layout of the bristles on this particular species. I guess I didn't explain that clearly enough. Maybe.
yeah. So one species has it one way, and the other one has it another way. And there was a mutation from one to the other, but we don't really know which way. But there's obviously more information because it can be one way or the other, in
Right.
one species or the other. But it doesn't match any of their ideas of deleterious net gain, net loss, and all of that. So
Right.
I think they have trouble understanding the question.
Right. Yeah. I kind of. I mean, that's. I guess that's kind of the point, really, because like they use the word information so widespread, or so universally that basically it becomes meaningless at this point. And I'm trying to
Right.
like, does it also apply? Because as far as I can tell, and I've talked about this before, that this specific bristle mutation, as far as I can tell, didn't seem to affect the fitness. Like the literature says it's a derived trait, but it doesn't specifically say if it's deleterious or not. And if we were to say, for instance, that it just changed the information, that it's effectively neutral, then according to them, they would still have to say it's nearly neutral because they still have to say part of it is deleterious, right?
Right. But there was, and then doesn't he ask a little? If we go a little further, doesn't he ask if it caused problems? I think.
That may have been the other video we went over. The one.
Okay.
This one was mostly SFT. The other one was NEF. I think. So.
But somebody was asking whether or not it caused problems in mating or decreased mating. So they were trying to. So when you said information, they wanted to relate it right away to an increase or decrease in fitness.
Right.
And
yeah, that's the problem, kind of.
It is. The thing is that between these two species of Drosophila, there is information in that there is a difference between one and the other, but
Right.
It has nothing to do with deleterious or beneficial or any of that at this point. It has to do with a variation. So for me, information is always a variation.
Right. Yeah.
You cannot when you assign positive or negative to information, what you're trying to do is assign meaning to it.
Right.
And that's a whole different subject. So I separate pure physical information, which is nothing more than differences or change, from meaning. And they
Right.
don't. They confuse the two.
Right. And I think that's what I'm kind of trying to address, is that you need to be more like specific with this difference between information in terms of fitness and what information they would, or yeah, you know what I'm saying? Like they need to differentiate between the two. Otherwise it's just confusing the problem, basically.
Yeah, they need to work out what exactly they mean by information. And of course they don't do that because for their purposes, it's the rhetoric that makes a difference. They don't
Right.
really give a shit about if we can work this stuff out in terms of biochemistry or molecular biology. And I think that's pretty obvious with this guy as well as most of the others. They just never think about it.
Which one, SFT or James Carter?
Both.
Both?
James.
Yeah.
Yeah, I listened to James Carter, and I really, really have a lot of disagreement with him on many things. He may end up coming on in a week or two, or I think maybe I forget what he said, maybe February or something. But we might actually have a conversation.
Okay.
But he's kind of
Yeah, that
new.
goes.
You know, there was Robert Carter, and then James Carter kind of came out of the blue. And I think he's written books, right? James
Maybe.
Carter? I don't know.
I haven't heard James Carter before until SFT came along.
Yeah. So he's kind of a new player as far as I'm concerned, but he's part of that creationist idea crowd. So
Yeah.
I do have some information on him somewhere. But we could have a good conversation, I think. So
Yeah, that'll be cool.
Yep.
Uh,
All right. I guess keep playing.
Yep.
And so when you eat pro-oxidant rich foods like fava beans, it can cause so much free radical damage to your red blood cells that the hemoglobin clumps up together, and in a microscope it looks like these ketchup spots. They're actually called Heinz bodies because of the scientists who figured this out. But it's a very common mutation, and it's not lethal, but these people are more resistant to malaria because the parasite needs NADPH in order for its life cycle. And so these people are naturally resistant to malaria, but they also undergo more free radical damage and oxidative damage because they don't make enough of their body's own antioxidant. So they actually call it disorder favism, because when you eat too many fava beans, it can negatively affect you with pro-oxidants that are inside of the beans. And so you can make up for it by eating extra antioxidants, taking supplements, eating more fruits and veggies for a higher external, your exogenous antioxidants. So in the sense, you could say that's a beneficial mutation if you're in a malaria laden, a plagued part of the world. But you take that environment away, of course.
So he's kind of off on a tangent, isn't he?
Yeah. I wasn't really getting what his point was.
Yeah.
Like, because he's talking about beneficial mutations and how it's a problem with sickle cell anemia or whatnot, because it helps with malaria, but it's a heterozygotic mutation. Is that right? Right. It only
yeah.
shows when it's certain carriers.
Yeah, I think. It has to be heterozygous, otherwise it causes issues.
Right.
And it was interesting because the population settled out mathematically according to what they had predicted with some pretty simple formulas. I think it ended up roughly about 50-50. It was kind of interesting, and stayed with the population because there were two pressures. There were two negative or purifying selections going on. If you were homozygous for the sickle cell gene, you would die basically. Right. And if you were homozygous for the other gene, you would die of malaria. So you had two different selection pressures, and they drove it to the middle, which is kind of interesting for population genetics. It's kind of a classical example, but I think Dr. Dan or somebody else might know more about that than I have worked out. But anyway, we'll go on.
Yeah.
They're not better off than the wild type.
Right. So I guess I agree with that. So for my question would be, for the example of an enhancer mutation that I gave, I'm kind of looking for the opposite in terms of I guess the first part of the question, because it's kind of a multi-part question, but the first part of it would be what specific part of that enhancer mutation I just gave counts as a loss of information, and why?
Do you know what the role of an enhancer element is?
Yes.
Yeah, because I think some people might be confused when you say enhancer. They're thinking this is a mutation that enhanced the organism.
Yeah. I guess I should be more specific and say regulatory element, perhaps.
Yeah, there's not every gene has these, but upstream of the main coding region that where the mRNA is going to be transcribed from, you'll have these various enhancer regions where certain proteins will bind and bend the DNA and make it easier for the two strands to open, and for the RNA polymerase to get in there and also recruit. There's several different proteins that can be recruited to increase the turnover rate to make these messenger RNAs. And so we call them enhancers because they enhance the rate of transcription, especially for very active regions where you need to get lots of protein made over and over again. So so your question about the enhancer was, is there an opposite mutation than what has happened in the enhancer region of the Drosophila DNA? I'm not sure what you're asking.
Well, I guess to clarify my question, so this was a single substitution, and from what I understand of the creation model of genetic entropy, mutations tend to cause a loss of information. So my question would be, for this specific substitution, what would the specific loss of information component be?
Well, it's like, mutating part of a paragraph, but you still get the gist of the paragraph. It's not as idealized, I guess you could say, as the original code or the original sequence, because it sounds like these bristles prevents as successful copulation. Is that right? So then they produce less.
Yeah, you see, he did ask about that, and I don't think that's the case at all, right?
Yeah. If anything, like I would, like I said, the literature that I looked at didn't say anything about its fitness effects. It did mention that bristle number on the sex combs of the forelegs in male flies helps them court the females. Like I think it's a way that they can grab onto them so they mate more successfully. So if anything, it would kind of sound like more bristles would be more useful, as far as I can tell.
Yeah, but they, because they're two successful species, so we can't even talk about loss of information at all. And
Right. That's another good point. They say it's a derived trait, so obviously it didn't cause extinction or anything.
Right. So nothing happened. So what was the first name of the guy in the paper again? Was it
Well, the one I remember is Nagy et al.
Nagy? Okay,
Yeah.
Nagy. Yeah, okay, because I couldn't find it. Nagy. There you are. Okay, I just want to pull up the paper just so I have it.
Right.
Okay, we'll go on. So he's trying to write. He's trying to shoehorn this, and he uses the same lame, which looks bad on him basically because he's supposed to be more of a professional in SFT, and he uses the same lame analogy to words on a page. If we start modifying the letters, after a while, you won't be able to understand it. But that isn't what you're talking about at all.
No, I'm not. I'm not talking about analogies or books or anything. I'm talking about this specific mutation. If you can't give me an answer, then I don't think you know what the heck you're talking about.
Yeah.
Offspring, are they totally incapable of reproducing? I'm not familiar with this mutation.
And the whole point, Andrew, I was going to just point at the fact that when it comes to genetic entropy and the eventual genetic degeneration of larger mammals, especially humans, it's the fact that most mutations are deleterious. And regardless of if you can get a truly beneficial mutation, like right now through the Lenski experiment, there's a paper that came out that suggests truly beneficial mutations are about 1 in a million. Okay, so they've done numerical.
Have you seen a paper that says 1 in a million? I don't know.
I've heard like Sanford's papers quoting it, but I haven't seen the paper directly.
The problem with Lenski is I think he's got about 400 papers out there. And I once tried to download them all, and I think I made it to like 30 before I got tired. I don't know.
Simulations were very generous, considering what you're saying. Let's give the evolutionists 2, 3, heck, give them 10 truly beneficial mutations that added information. I don't believe that those are out there, but let's just say they were, give it to them. The calculations have been done because the majority of mutations are effectively neutral. So they have very subtle effects on fitness. Natural selection essentially can't see those mutations, okay? So they get passed on from generation to generation. Now, the majority of even your beneficial mutations are reductive and functionally compromising to the organism.
Okay. So they're neutral mutations, but they're reductive.
Right.
So he kind of slid one in there. He can.
Yeah, I mean, like, I haven't really understood this nearly neutral thing. Like, how can a mutation be effectively neutral but still have slight deleterious effects? Like, do you know what that's about?
It doesn't make sense, actually. And that's the problem. And they think it does because they use their analogy that if your car is rusting, if it has 1 spot of rust, not a big problem. But if you have like 1,000 spots of rust, then it's a problem. But when you look at actually what you're talking about with mutations, you've got to say you have some area on the chromosome and it's a neutral mutation,
right? And
it mutates. That thing will never, ever have an effect on any organism. It is completely neutral.
Right.
And then there are some that don't even change the amino acid. But I mean, in some cases, the mutations do not matter at all. And then in other cases, they may make a difference, but it's going to be a small difference, and it could be as easily positive as negative if you're talking about just like, say, a gene mutated, had one mutation in it, a protein, and that was in an alpha helix, and the alpha helix still folded as an alpha helix. It would not make any difference. So then if another one happened and another one in the same gene, the problem is that these mutations don't hit that same gene except once in every a million years or so, and so, it's like, what the hell would it be like? Well, needle in a haystack kind of thing. It's because, but he kind of implies that if there's one mutation anywhere in the genome, that the next mutation is going to add to that. That's going to be additive, and that's simply not true.
Right.
And it's probably not even true if it's in the same protein. But say this alpha helix, say you're really unlucky and mutation after mutation hit that alpha helix, and then we get a switch to maybe a proline, and all of a sudden we don't have an alpha helix. And if that alpha helix was important, that organism will die right out of the box. And that's it. It's over. That whole line's gone. So,
yeah.
Hey, Walker.
Hello. Yeah, y'all convinced me. I don't know. I like talking about nearly neutral theory. I think it's interesting.
Oh no, sorry about that.
Genetics and stuff. Huh? Oh no, you're good. Yeah.
Yeah. Okay. So Standing is coming in here with his bolt. He's sticking his ShamWow salesman routine in here. So.
Mm.
Well, let's just say the example you're providing, let's just say that's truly a novel example of an information gain mutation. Let's just say that. Well, those have been considered.
Okay. No, did you see what he said? An information gain. So he's assuming that you're saying information is beneficial.
Right. That's I didn't say that at all. I wasn't talking about whether it was novel or whether it gained information. That's the whole thing I'm trying to figure out, is this information definition thing. And he obviously means that.
For them, yeah.
Yeah.
But Walker, did you agree that, like, when he talks about accumulating mutations, they would all have to accumulate on the same regulator or the same RNA or the same gene?
At the very least, his model requires a linear accumulation of mutational, like, additive effects.
Additive effects.
So not necessarily the same gene, but every single genotype needs the exact same fitness for your relative fitness to stay the same while your absolute fitness is declining. And that's a stupid idea.
Yeah.
Right. Well, like, that isn't fitness relative in evolution?
Well, so there's a distinction between absolute fitness and relative fitness.
Okay.
So absolute fitness is basically just the proportion of one phenotype to the next phenotype across generations, whereas your relative fitness is adjusted for the most fit phenotype.
Yeah,
right. So you can have
you're comparing you're comparing one allele to another. Basically.
Yeah. So you can have an absolute fitness of 1.5, right, which means that the proportion, the frequency of that allele grew by 50% in one generation, which is pretty absurd. But you would adjust that with the other allele, which, let's say that other allele didn't experience any change, right? So its absolute fitness is 1.0. And then you would divide them both by 1.5 to find the relative fitness of each. So the relative fitness of the most fit allele is always 1.0, whereas the other ones are like, for that example, it'd be 0.66.
I wanted to mention Scott Duke said that idea of especially humans are degenerating. Did Carter say that? I think he may have.
It may have been. I think he's probably quoting SFT if I had to guess. And
yeah, and it's about the fall, because they have this fall thing and they're so bent upon that.
I think using humans as the only example is probably a crappy example because we're the exception to the rule.
Yeah.
Because humans obviously are experiencing, weaker purifying selection forces. Right. If you have a some sort of mutation that would otherwise be pretty crippling, and you likely wouldn't have offspring in a hunter-gatherer society, it probably won't affect your fitness too much, like cystic fibrosis or something. Like if you had cystic fibrosis, way back when, you would die. But now we have treatments for it, and you're probably going to live a relatively normal life.
Right.
Yeah. Is Otangelo Grasshole talking about Erica? What the hell? Anyway, what you talk about? Okay. Yeah. They love to talk about humans because you can make up all sorts of bullshit about humans, like autism and increased dementia and increased cancer. I went and looked up the stats on all those things. And it turns out cancer's been decreasing since 1989.
Oh, wow.
And you know what? It's odd, but that was right around the time that better mufflers, catalytic converters, and non-smoking both came into our society, right?
Mm-hmm.
And so cancer's going down, except for there's two of them that are staying the same: prostate cancer, and that's that would stay the same because the longer you live, the more prostate cancer you're going to get. And the same with dementia and Alzheimer's. The longer, the more we extend our lives, obviously, the more cases we're going to have. So they make up these statistics and they never actually go out and look at them. But when cancer statistics, for one thing, fewer people are dying of cancer because of better treatment.
Yeah.
But I looked at the rate of cancer itself, the rate of people that were getting cancer on the American Cancer Society website. And all of them went down except for, I think it was prostate. May have been lung. I'm not sure. No, I think lung cancer went down. There was one other that one, stayed the same.
I would assume lung cancer is decreasing.
Hey guys, guess who's in the chat?
Oh no!
It looks like another person. People keep thinking that Otangelo Grasshole is the actual Otangelo, and I find that very amusing.
Well, I think he actually is I think it's Otangelo on a bottle of wine. That's my theory. Is that when he drinks, and he frequently drinks after our weather reports because we're so tough on him. So anyway, yeah. My brother, I found an atheist willing to debate you in intelligence. Yeah. Okay, Standing. You might want to come in here and defend yourself because we're ripping you a new asshole.
Yep.
Go on.
Sure. Thanks for being here, Walker, by the way.
Oh, yeah, of course.
The points are always good.
So, yeah, I like talking about population genetics. It's actually what I'm going to be researching next semester, so that'll be fun.
So, Andrew, do you have a problem with our safe word foreskin? Or
is that related to like sexual stuff?
No.
No, not necessarily.
No.
Okay, I'll try and use it. I thought it was.
Well, it refers to something on your penis, but that's not necessarily sexual because our anatomy isn't necessarily sexual. But if you want to think of another safe word, I'd let you use it. But we got to have a safe word because your voice sounds the same to me on the video as it does.
Right.
And I'm assuming you weren't throwing the word foreskin around a lot last night or whenever this was.
Right.
No. Yeah.
Just say
I'll use it.
Or just say skin or whatever, but say some goddamn thing so I can pause the video.
Yeah, I'll try and use skin.
Okay.
Skin on my hair.
I'll try to get used to that, but I'm going to be 70 day after tomorrow, and I don't know if I can get used to anything new, but I'll try.
Right.
Old dog, new tricks. No. Okay, moving on.
Numerical simulations. They've thrown in any parameter you can think of. They've even worked it out.
He's talking about Mendel's accountant. And I downloaded that and I ran a scenario and it came up with some really kind of fucking ridiculous. For one thing, the parameters that you put in there were really suspicious. And have you used that?
So no, I haven't. But there's this one guy on r/debate evolution named Swery Biochemist, and he said that he tried running the Mendel's accountant using the exact same parameter, basically using the exact same setup of Richard Linsky's experiments, and he couldn't get the results that Linsky had. There was still error catastrophe, even though we know for a fact that there's obviously, Linsky's populations are still doing fine.
Right.
It's pretty funny. And he said that like to even get a stable population at all, he had to set the beneficial mutation rate to something like 95%.
Yeah.
Which is absurd. Like,
and then I looked a little bit into the code on one of the older versions. I think that was written in C plus. I can't remember. It might have been written in C, and it just looked suspicious what they were doing. And now they have a new version that's in I think either Go or Erlang and Python. It's a combination of the two, and I haven't yet really taken a look at it. But when I ran it, I thought it was so weak, and so the parameters, there weren't enough parameters. You couldn't control them enough. I forget what it was, but we'll run it sometime on this channel and see if we can figure it the hell out. Maybe Standing for Truth, who knows how to use it, uses it all the time. Maybe he can come in and show us how to use it. So
yeah,
we can
I'm
use it.
sorry to say, I'm sorry to say, Standing, if your simulation doesn't match reality, then it's just wrong.
Yeah. Yeah, Mendel's accountant, as far as I can tell, is a piece of shit. So
yeah,
until somebody can prove otherwise. And nobody else uses it except Sanford in the creation. Well, in fact, only Sanford uses it.
Yeah, I was about to say, I think that's telling.
I think it is. Yeah.
If you
that the majority of the
Should I go?
No, I wasn't going to say anything.
Okay.
genome is junk. Okay, and these papers have been published. You can check them out. And still, the outcome is always the fact that genomes degenerate, even with
Hold on a
build.
second.
They've thrown in any parameter you can think of. They've even worked it out that the majority of the genome is junk. Okay, and these papers have been published. You can check them out. And still, the outcome is always the fact that genomes degenerate, even with a huge amount of purifying selection, even with truly novel beneficial mutations. The genetic degeneration can't be stopped. Here, this is why the key question is net gain versus net loss. So that's the point, is you're not going to need 1 or 2 or 10. You're going to need thousands and thousands of truly beneficial mutations to counterbalance the damage. So that's
just bullshit.
Right. If I had to guess, there's got to be some sort of like just very basic population mechanic that like Sanford missed in his programming. Because that's just not how reality works.
Right.
You know, if you have an infinite population size, there's no such thing as a nearly neutral mutation. So you can't even say that mutations accumulate. So and most of these, population models and stuff are done assuming infinite population sizes. Because that's what like
I think they have a fixed population size. I think I ran
really on Mendel's accountant. Okay.
Yeah. And I think I ran 10,000. If you run simulations on like 100, you're going to end up with a lot of problems. But I didn't see a parameter for specifying junk DNA.
Yeah,
I don't know what he's talking about. We will pull that up here at some point.
Yeah, I would love to take a look at it. I don't know how it would work, but I don't know. I've just not heard the best things about it.
Who came up with it?
Mendel's accountant. It was mostly written by Sanford and a couple of other creationists.
Yeah, they had some programmers that they. One of them died of cancer. The one that wrote the original, or no, he wrote the new version. I think that's why it hasn't been updated, and probably nobody knows how that program works. It's out on GitHub. In fact, I have it here on my. I downloaded it, but what was I going to say? I will bet 350 that Standing has never used that program, never even downloaded it, looked at it, and probably doesn't know where to find it. He's just going what his buddies tell him.
Well, Standing is right, though. Like, you can use whatever parameters you want in Mendel's accountant, and it'll eventually lead to, like, mutational meltdown.
Yeah.
It's just that's not reality, right?
Yeah, right. Like I said, if your simulation doesn't match reality, it's wrong.
Yeah.
That's a good point. Okay, moving on.
The bigger picture that I'm trying to convey, Andrew.
So if I'm getting that correct, I guess like when a net loss have to be a loss greater than a gain, for instance, is that what you're saying in terms of information?
I guess sometimes information can be altered, but you still end up with the same product. Like if a codon for glycine changes from GGC to GGT, you still end up making glycine. So from the protein perspective, it doesn't change anything, but technically you've changed the code, the original information, but it didn't have any detrimental.
He said a change in information, right?
Okay, once again, that's I've argued against that a lot, and I think it's really dumb to say that literally synonymous mutations are deleterious.
Yeah.
Like
Right.
the word synonymous is in the name of this type of mutation. You know, it doesn't do anything.
Yeah, they could
It's
be
effectively the same.
It could change things if that particular amino acid was less available. Or no, it wouldn't
No, because it's the same amino acid. Yeah.
Okay.
But
Okay.
I've heard them claim multiple times that all mutations are deleterious, including synonymous mutations,
Right.
because it's like you're getting further from God's intended script.
What I meant to say is
how do you even argue against that?
What I meant to say is if that particular tRNA was less available or less stable or for some reason. I think there are very slight differences.
Well, so when you're mutating the third base of a codon,
The same tRNA?
Yeah, you're using the exact same tRNA because tRNAs have this special property called wobble base pairing, where the third base is usually pretty flexible. So usually, you know, if you have a purine exchange, you know, the tRNA protein is not going to be able to tell the difference. Well, I guess it's not a protein; it's whatever they're called. But you know what I mean. Yeah, so the wobble base pairing means that you're using usually the exact same tRNA.
Yeah. Okay.
Ribozyme. That's the word I was thinking of.
Ribozyme. I think there are some. I think there are some differences, and there are more than one tRNA that will match those things. But I have to look into it.
Well, there probably are. It will likely depend on which codon we're talking about. But like, there's some codons. I think like lysine. There's like 4 ways to make lysine. There's probably 2 mRNAs or tRNAs that they use.
Yeah.
Right. And it'd be 1 for each of the variable second positions.
Yep. Okay, so I'm going to move on a little bit.
In fact, there was a silent mutation.
That's a good point.
No, it ain't a good point. It
It
doesn't.
would be like this, Andrew. It would be like
leucine,
climbing a mountain, and
here we go.
You know,
yep.
You're taking a bunch of steps backward, but once in a while you take a step upward. But then you take another 10 steps backward. Then once in a while you take another step upward. You're never going to climb that mountain, or you're never going to get rich by losing money. You know what I mean? And that's how it is in the genetics, where there's really no amount of benefit.
He calls it the genetics. You know, the genetics. I was reading the genetics the other day. And anyway, Standing for Truth has this idea. It's a difference between discrete and an additive whole. You see what I mean?
Okay.
Like, okay, he sees it as climbing up the mountain or falling back down the mountain. And actually, that's not what happens at all. We're talking about the shoelaces on the climber's boots. We're talking about what color shirt he has on.
Well, I would say in some ways it is additive, but it's not linearly additive.
But how is it additive?
Well, because when you're looking at selection, like acting on a population, it's all of the different mutations affecting an individual, right? So like it's somewhat somebody's differential reproductive success is based on the cumulative alleles that it has. If that
okay, so if he both had a weak chin and he had ugly hair at the same time, he would mate less. So there would be, so you could say those are two mutations, but those aren't really mutations; those are variations. And it's so simplistic.
Well, yeah, I would agree. I think it's overly simplistic. Like, even though I would say it's additive, at least on the individual level, whenever if 2 people have weak chins, their chins aren't the same amount of weak. So you're always selecting against a different phenotype.
Yeah.
Does that make sense? So like,
but I'm not looking at this at the phenotype level. I'm looking at it at the gene level.
Yeah, and I think that's when you're talking about selection, you're ultimately talking about it at the genotypic level. But the phenotype is what's expressed. But that's what I'm saying, right? So it's not like, these mutations aren't having the exact same fitness, increase or decrease in every single individual in a population, across every single generation.
Right.
That's dumb. I mean, mutations are fairly stochastic, so you can't really predict what any specific mutation's selection coefficient would be. And when you add up all the different selection coefficients of an individual, they're going to vary across populations. That's inevitable. And when you have this variation, there will be selection.
Yeah, but the thing is that if it is truly additive, then it will truly be purified out of the population.
Yeah, that's what I'm saying.
Across that threshold, and that's what Dr. Dan was talking about with that threshold. It enters this equilibrium.
Mm-hmm.
And
the other important part of the equilibrium is that there's variation across the population. Because if there was just a hard threshold and all of the population was accumulating these deleterious mutations at a constant rate, and they were having a very constant fitness effect, then there would be just a breakdown.
Yeah, and on the somatic line, you have the same, you have the same amount of mutation. In fact, even more so because of UV light and all that stuff. So on the somatic line, how many times do certain tissues divide? And if his idea was correct, then you would age so rapidly that you wouldn't ever make it over 3 months old. I think if these deleterious mutations collected, that sort of thing. I don't know. Doesn't make sense.
Everybody carries some amount of deleterious mutations, and everybody inherited deleterious mutations from their parents because that's what, whenever Dr. Dan talks about like a fitness equilibrium, we've saturated the equilibrium essentially, where if we get more deleterious mutations, there's going to be selection against our genotype. Whereas if we have beneficial mutations, there will be selection towards that. Does that make sense?
Yeah. Slight change in a protein coding region may influence the mRNA stability, and that may influence protein fidelity, but it's rare for that to happen. And even so, it's definitively deleterious. The thing is, if you have a vital function, if you have a protein that is absolutely vital, that you have to have on either one chromosome or the other, and if there's mutations in that and it doesn't work, then you got a dead organism. So you've got instantly, you fall all the way down the mountain and you crash in the valley and you're dead. So he sees, he says because they're near neutral, he doesn't think that they can add up so that purifying selection can take care of them, or he thinks they're immune to purifying selection, but that's not the case, because if they are additive, if they do add up, then purifying selection is going to get them.
Right.
And he sort of like, it's like he's playing, what is that? 3-something Monty. What the hell's that called? Yeah. Where you put the bean under one of the cups and move it around. What do they call that?
I don't know. The shuffling game.
Something Monty. I don't know. Yeah. Somebody in the chat will know. But anyway, it doesn't make any sense. Okay.
Dr. Dan's in the chat. Oh,
who is?
I think he agrees with you, Speed.
Somebody agrees with me.
Yeah,
selection will operate.
Yeah, there we go.
Okay.
Yeah. And that's what
I love when people agree with me, Dr. Dan. Makes my day. Okay. So moving on.
Sure.
Beneficial mutations that have been observed that can truly counterbalance the damage done through these low impact near neutral deleterious mutations, even these essentially silent mutations. There's papers out now about translational pausing, how the third position codon variations act in a functional means to slow and speed processes in the cell. So it's kind of like your tire, and the treading on the tire it's slowly wearing down where it's almost not noticeable. Someone could say, however much.
Yeah. Okay, if Standing for Truth is still in this chat, I want him to answer if these nearly neutral mutations affect the relative fitness of the population, and if not, why not? Or like, I guess the relative fitness of a genotype, and if not, why not? Because if you're claiming they're additive to the point where they're affecting reproductive output, they have to affect the relative fitness of an organism. Unless you're saying that they're neutral. All yeah. And at that point, if they're affecting relative fitness, like you said, they're not neutral. Unless he's saying that all genotypes are equally decreasing in absolute fitness, which wouldn't change their relative fitness. And that's, once again, that's a dumb proposition. That's like saying all of the mutations have the exact same fitness effect across all generations, across all time, like across the entire population.
Right.
That's not going to happen. It's unrealistic.
What catchphrase? What is the new catchphrase? I think it's an old. He come up with something new. Standing come up with something new. Anyway, moving on.
Yep.
Oh, translate. Oh no, translational pausing. I guess Standing hasn't used it. John Maddox uses it all the time. And like I say, translational pausing doesn't appear to be a big deal in the grand scheme of things, but they keep bringing it up because that would mean that that third codon could have some sort of an effect because the signal wouldn't get hit for the translational pausing, but there's no actual evidence. Well, maybe there's some evidence of that. There was one organism that they looked at for this translational pausing. I guess we'll have to dig the papers out and take another look.
Yeah.
That's something John Maddox has been harping on for 6 months now. So anyway, ribosomal pausing. They really think it's an ace in a hole for some reason. Interesting.
All right, now watch when
is neutral to the car. It had absolutely no damage or effect at all. But over time, the treading eventually will wear down, where it's damaging, and you got to replace the tire. So even in these redundant codons, for example, enough mutations to them affects the outcome of the organism in a negative way. Is my point. Go ahead.
Right. So I would agree with the redundancy. I guess, like my question would be, you were saying with your analogy of the mountain, the 10 steps going backward and 1 step going forward. My question is,
brilliant.
In terms of the example I gave, what is the steps backward? I guess, like in terms of that.
Right, and that's my point. Is this has all been done and calculated, as in, hey, let's just give the evolutionist a few truly beneficial mutations. So let's say, for sake of argument, okay, that example you're providing, let's just say there truly is no loss of some kind. Let's just say it's not reductive. The point is this has all been done through numerical simulation, and even with a generous amount of truly beneficial mutations that we don't even observe, they're just being generous for the sake of it, just to see what the outcome is.
Okay, so what is he saying? That he's saying that because they have this program and this program found this result, then we've proven it.
Yeah, I still find it funny that he's saying that the reason Mendel's accountant doesn't match reality is a good thing.
Yeah.
Like, that logic kind of blows past me a bit.
Yep.
That's not how science works.
Yeah, we have this computer simulation that, empirically does not match how the world works. Therefore, evolution is false. What?
I mean, like you can have a simulation that does match reality, but that's completely different. Like you can have galaxies simulated, but that's like that's because they do match reality there. Like the galaxy simulations, they match the distribution of galaxies, I believe.
Yeah, that's the other thing because of selective sweeps. There, if it does occur, something does occur, it will wipe out 99% of the deleterious mutations that you have rusted your car.
Yeah, if you ever have like a mutation that's like super beneficial, it'll actually reach fixation relatively fast. Obviously, It depends on a number of factors. But.
Yeah, but then they pull in this epistasis. Am I using that right? Yeah, I am. Basically, two genes interfering with each other, or positively or negatively. They always pull that out, but then they never really think about that math either.
Yeah, they also know recombination is a thing. Epistasis can act in short terms. It can lead to like a short-term fixation of deleterious alleles. But if you're looking at a reasonably sized population and these genes aren't directly next to each other, it's not going to have that big of an effect.
Yeah, the only reason it's going to act is if it has a positive effect, and it pushes the population along those lines. So,
well, the way, correct me if I'm wrong, obviously, the way they use epistasis is to basically say like, okay, well, if you have a beneficial allele, well, it's surrounded by deleterious alleles. So whenever you start fixing that beneficial allele, it's also dragging along all the deleterious ones, right?
Yeah.
Yeah, but like I said, that's dumb. We have recombination.
But
over long time spans, it matters.
And by the same logic, if those surrounding deleterious mutations have an effect, then you got a dead organism, or you have something that reproduces.
You wouldn't even have a dead organism if there was if this beneficial mutation is so good that it outweighs the cost of those deleterious mutations, then you definitely don't have a dead organism, right? You still have an organism that's more fit than the original population.
Right. God, could that explain Trumpers?
Epistasis.
Is that why they're up to nearly 50%?
Like a point I might make is that he's saying that like they're additive, right? Speed, the like one isn't noticeable, but it adds up over time. Well, what's the difference between that and it not having effects whatsoever? Like if it's not even noticeable, like at this stage, then like, can someone tell me the difference here? I'm this is what I'm trying to understand.
Well, the thing is, you've got to hash all this out at the molecular level. You can't look. You can't be thinking that one gene is going to like have this particular effect on the phenotype and then a negative effect, and then there's going to be a positive effect from a gene that's epistatically linked to it. Right. You have to look at the molecular level and see what the hell is really going on, because there are so many other possibilities other than there are going to be two distinct phenotypic features hooked to one, two genes. I mean, it isn't that goddamn simple, so, and that's where they fail, that they never, do you know what I mean? You have to hash this out at the molecular level, because a lot of these mutations aren't going to make a difference.
I agree that's, obviously ultimately biology, but you don't have to hash it out at the molecular level because it fails at the macroscopic level too.
Yeah. I don't believe in the macroscopic level. It's a problem with studying molecular biology. You quit believing in the macro. Also, it's like, fucking explain it to me with molecules, damn it, or don't exist. What do we got here? And also, if you're stuck with the fixed bad alleles, you wipe out all the other bad ones in your organism that aren't linked. Yeah. And you can wipe out a good one with a bunch of bad ones, I mean, most beneficial traits don't actually make it to prime time,
Oh, for sure.
Yeah. And yet they're still really, or they're not common, they're rare, but like they're still abundantly obvious.
What is a holy prepuce?
Prepuce?
It could be like foreskin.
If you assume that beneficial mutations occur like 1 in 100 mutations, that means everybody is currently carrying a beneficial mutation.
All make worse.
So
all mutation make worse. Otangelo Grasso, I believe, has a PhD in something. Okay. Go on.
Sure.
Yeah.
Yeah, I think it's almost done.
What day is it?
Monday.
Oh, fuck. Oh, I'm so happy it's Monday.
Oh, really?
Yeah.
Wish I could say the same.
I would have missed Ian if it was Tuesday. Okay.
And the outcome is still always genetic degeneration. That's the point, is even if this example you're providing truly is novel and it's truly a step
upward, that's not what I was saying.
Million deleterious ones, which means that one step upward, if it truly is that, it ain't going to counterbalance the other million that are downward. You know what I mean?
But at the end of the day, it's still a fruit fly, just like a human with more hair on his back compared to a human with less hair on his back. There's no
Okay, wait,
change. It's
foreskin.
just a micro variation of the same kind. So
Foreskin.
how is that? How are those extra bristles on the leg
Wait.
of the fruit fly
Oh,
going
are
to
you saying
result
something?
in
Oh, I
something?
said
that-
foreskin.
Oh shit! I was
Oh
typing.
no, you're good. He didn't say anything novel. Okay, I was thinking about this earlier. This is the most nails on a chalkboard argument to me, because yes, like it's nothing other than a fruit, and it's nothing other than a fruit fly. In the same way that we're still eukaryotes, or like we're still mammals. We never stopped being mammals.
Right.
In the same way that we're descended from our parents.
Yeah.
We never stopped being descended from our parents and inheriting what they had.
Yeah, we never stopped being apes, right? Like, oh, well, it's still just an ape. Okay. And any of our descendants are still going to be humans. They'll just be, like, ultimately, I would presume, different varieties of humans.
Right. And this is the problem with it, because, like, generally, what we consider novel is just really just under the surface a modification. Like in a broad sense, like it has new emergent properties, but it's almost always based on what came before.
Yeah, like I said, that argument is just one of the dumbest possible arguments, because I think if you've shown that any sort of mutation that meets their gain of function beneficial mutation like definition, they'll just dismiss it as like, oh well, it's still just X or Y.
Yep.
But if you can have, like, by your own standards, beneficial gain of function mutations, then there's no, like, problem with evolution, because evolution doesn't ever say that a dog turns into a cat or a pine tree turns into a whale. If that did, that would be magic almost. Like, I guess it's theoretically possible, but like so unlikely to the point that it's just worth dismissing.
He's a wonderful distraction. 3 sequence. Yeah. But I wonder, muscular dystrophy is what is it? Lamins around the nucleus. It's interesting. There's an effect on the lamins that hold the chromosomes basically in the regions in the nucleus, and I think it's muscular dystrophy. But it only affects the muscles because apparently it's too weak to hold it to the nucleus, to the nuclear membrane. So if you move your muscles around, because muscles are always, there's a lot of force on muscle cells, and apparently it fucking breaks it up and starts killing your muscle cells, which is interesting. But it's I think it's a single mutation in lamin, I believe.
Well, lamin looks exactly like a cross. So
yeah,
there you go.
Yeah, but there's like one mutation that will just screw it up, and it doesn't work quite as well. But it's interesting that the muscles are the first things to break down in the body. It's because you can imagine something getting stretched, kicked around. You know, like when you work out and your muscles feel like shit. There's a reason for that.
Yeah.
Yeah. Lots of work. Anyway, okay, moving on.
Yep.
I'll try to listen for that weak little foreskin you guys muster. Like if you say foreskin, then I'll hear it.
All right.
I did say foreskin.
Yeah, but I was typing. I wasn't here. I was
this
asking.
is an exception to the rule.
Okay, sorry. Yeah. Okay, which proves evolution false.
It's a non-fruit fly.
That's
Right,
the point.
but
It doesn't
that's-
change the structural. It doesn't add new structural design
Oh, and here we go.
Exactly.
body
It's just a
plan,
micro variation.
and it doesn't modify an extant feature so that it takes on new biomechanical function. And
That's not what I was arguing.
It doesn't participate in the idea that organisms morph into fundamentally different types of organisms over time.
Okay, wait,
It doesn't
foreskin.
participate in that.
After like 12 years on YouTube, does Neffy
Yeah.
still think that we think this shit's like Pokemon?
Yeah.
Morphing into a different species? Come
Yep.
on, dude.
Well, they were talking on a stream once, and I think it was in one of Nephilim's streams. Maybe it was Praise. But they were actually talking about, what happens. A fish, all of a sudden runs out of food, and he decides to climb on shore, and he goes a limb. And I mean, they were actually saying stuff like that. It was late at night.
What?
And I have
Your
a bookmark.
dog is evolving.
Huh?
I said, what? Tiktaalik is evolving.
Yeah, right. Yeah. It sticks its head out of the water. And John Maddox was saying, the most, and they said the most stupid things about, fins to limb evolution. But for Nephilim, he talks about, he makes a difference between, what is it, anatomy and morphology, which is really kind of fucked up.
Actually,
yeah, he makes a distinction. Yeah, I think he calls anatomy if a limb grows longer, and morphology would be if you added another bone, or I don't know.
What?
So does this mean that we have the exact same morphology as chimpanzees? He wouldn't want to admit that, right? The changes between us and chimps are only anatomical, according to him, and not morphological. Yeah. So they can happen.
I wish I have that bookmark somewhere. I'm gonna have to go dig that up someday. That was a wonderful stream. They said about the dumbest things I've ever heard, but they were talking amongst themselves. And Praise was also talking about his idea that all reality is in the mind at the same time, and they were mixing it with a bunch of evolution. It was funny as shit. Anyway.
Jerry Penna said it was 3-card Monty.
Yeah, okay, 3-card. What do they call it? 3-card? I thought it was like a bean under a couple of like 3 glasses or something, and they move it around real fast.
Well, I think it's because originally they used like cards, and they would shuffle the 3 cards around.
Yeah,
and you had to find like the ace of spades or whatever.
And they bark at you the whole time they're doing it to distract you, and that's exactly what SFT does.
God, did they actually bark at you?
Yeah. They're talking to you the whole time. Now you see, they're just doing this whole rap, just like SFT does. I wonder if he came from the carnival. That's maybe, yeah.
Conspiracy theory level there.
Well, check that one out. He might actually be a carnival worker and not a ShamWow salesman. But okay, moving on.
Yeah, I think it's almost there.
I think we're done.
Yeah, I feel like he was gonna say something else, but I'm not sure. So maybe going for a little bit, but I think
we'll play for a few minutes. Let me know.
Process. Then it can't be said to be inducing any kind of evolutionary change.
If you look at the rate of these so-called beneficial mutations and how many of those you would need before another system develops that would somehow create a distinct species, that rate is entirely too slow.
Really?
Can I ask a question?
Yeah, go ahead. I got to get going.
All right. First of all, there, I think that's where I stopped talking.
That's it.
Yeah, that's pretty much it.
Okay, Otangelo Grasshole, you just went too far. I have bigger one than that.
Oh God, what was I going to say? Oh, there's not enough time. Like I said, I don't think people can conceive of how long like 1,000,000 years is.
Yeah.
No, I can't.
I for sure cannot.
There's a guy named Lauren Isling, Lauren Isling, I think, that did a book back in the 60s called The Immense Journey. And he talks about time, and he does it poetically in such a way that when I read it back when I was a teenager, it actually gave me the feeling of understanding for just being able to glimpse the immensity of time, millions of years. And it was really cool the way he put it. It's called The Immense Journey. I think it's Lauren Isley. I just bought the book somewhere. I don't know. The other problem with buying so many books, I don't know where the fuck my books are. And I don't have any room anymore to move around in my room. I just can't move. You know what this is an example of? This is
entropy.
Yeah, fucking genetic entropy in my room. I keep adding little bits of books to my room, and pretty soon they're all going to fall down on me and kill me and my cats, and that'll be it. And then they can ship them off to midnight.
You better build a new house, Speed.
Yeah, I thought about, if I had a lot of money, what kind of house I would build, and it would be about 3/4 library, about 1/4 living space.
Yeah, that would be awesome.
Yeah,
the library of God, I forgot what it's called, the one from Avatar.
Well, you could have like shelves, like library shelves with certain sections with real high shelves, so you can put the washer and dryer there. and then you could put like a cutting board.
You know what would be cool if your house was a library, you could have secret passages connected to books, and you're saying like, here, you want to see my laundry room? Here's my laundry room.
Yeah, you could just put secret doors all over the place.
Yeah, exactly. Yeah, slide away in there. There would be. Need to use the bathroom?
Oh, it's the book on the right.
Yeah. Yep. Go get my copy of Lewin's Jeans 10th edition and just pull it out.
Mm-hmm.
Yeah.
Well, I think it's safe to say that this discussion with SFT was surprisingly less enjoyable than with Neff.
Really?
Really?
I've always thought that SFT is pretty like fun to talk to generally.
If you can get him one-on-one talking without him doing his spiel, he's he's kind of fun to talk to.
Love to have a one-on-one conversation with him about population genetics. He asked me the other day like to have a debate with him on out of Africa, and I feel like my arguments would be about the same as they were for Neanderthals. I mean,
he always wants debate because then he can do a sales pitch. But if he comes to our channel and has one of those conversations with us,
yeah. And I would just want to have a conversation about like the population mechanics behind genetic entropy, and not even like debating genetic entropy, but just explaining like, here's current population genetics. Here's like the idea of what relative fitness is. Here's what a selection coefficient is, and here's why, like, if you accept that these principles, which are, they're just mathematical principles. If you accept that they're true, then genetic entropy can't happen as just a byproduct of selection.
Yeah.
Right. Well, what do you think, guys? Do you think I got SFT on the bristle question like Neff?
I think they didn't take it down the information aspect as much as Neff did. Neff tried to.
They haven't even started to answer you. They didn't even begin to answer you on any. I think Nephilim probably tried harder than anyone else, but he didn't even. They just blow it off because they don't know about it.
Correct me if I'm wrong, but the point of your question is more or less to get them to like say whether or not something is a gain of information, or like a specific real world example is a gain or loss of information, and like why, right?
Like I'm not trying to say this is novel or this is not novel, because I know they're not going to argue with that, sorry, they will argue with that. But like what I'm trying to do is go the other route and get them to define the loss of information so that we can define information in general, which obviously they can't do.
It's a very noble task. But yeah, I think Neff at least tried to answer your question. Standing sort of just went down, well, you see, nearly neutral theory.
Yeah.
And that wasn't what you were asking.
Yeah.
Right. Yeah.
You were asking
good so far.
Huh?
I
think the question is the question is definitely proving its worth, I'd say.
Yeah. Sorry, I'm not trying to take the spotlight with this right here. I feel like I'm getting up there with you guys.
You have been up here for a long time.
Yeah, and also, most of the stuff that I'm talking about, I've learned in the past year. It's not like I have any like super deep background in this stuff. I'm a second year in college. Like I'm going into my fourth semester in like 20 days. It's not that deep.
What's killing me is that I'm all over the place. Like in one day, I'll study 5 subjects and then I reread something and I've got highlights all over the whole fucking chapter and I can't remember any of it. And it's because I'm not really working as a student on it, I guess. I don't know. I just read it and enjoy it.
Well, it's also the power of being 20.
I don't think that makes a damn bit of difference because I'm learning faster now than I've ever have in my life. As far as actually remembering terms and being able to recall stuff, and it's a lot faster now because I learned a few things about how to learn. And I'm better at it than when I was a teenager. So I don't think age has anything to do with it at all. The problem is that I'm fucking ADD as hell, and I'm trying to be a Superman. I'm trying to learn population genetics, bioinformatics, genetics, molecular biology, fucking geology, and every now and then Leo drags me into physics, and I've gotta pick something.
Mhm.
Yeah.
And then even with genetics, I mean, I can drill down into nucleoporins I could spend the rest of my life just studying that one thing. So what do I want to do? Do I want to keep skipping across the top, or do I want to drill into something? You know?
Yeah. No, I feel that. I don't know. I've sort of this past semester realized that I like the more like theoretical and mathematical sides of like biology. I just think it's interesting.
Yeah. Hey, Knock, you're here.
Hello.
Hey, hello. This is weird. Is everybody in StreamYard?
What do you mean?
All at the same time?
Yeah.
You mean there's shit going on out there I don't know about?
Well, I have this weird program for StreamYard, and right now I only see speed of sound in myself, and I'm like, how is this happening? I am really dumb.
No, we're all in the same call.
All right, so obviously, I think I need to restart my computer. So I'll be back. And when I come back, just know that I am the dumb person listening to smart people.
No. I have dinner waiting on me, so I probably need to get going.
All right.
All right. Well, anyways, I'll talk to y'all later.
See you, Walker.
Have a good night. Because I came in and I was like, man, y'all are really smart. This is so weird. I'm not sure. Like, I don't even have my like little hack button to where I can turn my little hack off and see StreamYard normally. This is really odd. I will be right back. I'm going to restart everything and I'll be right back.
All right. These dudes, enjoy the convo. See you later.
Yep.
See ya.
So we got to drill into this structure. What I want to do is you take the entire genome, right?
Okay.
And you take a look at percentages, and 50% of it is repeats, and 27% of it is introns. And I want to look at this concept of junk DNA, and I really hate the term because I don't see it as junk at all. I see it as future stuff that's going to be cool.
Right. I looked this up on Talk Origins, and like it says that the term junk DNA was like here, let me bring it up real quick.
It isn't used that much. Like in textbooks, you could hardly find anything on it at all.
Right. Like it said something. It was like it was used even when we knew that the rest of DNA had a function, or something like that. Like it wasn't. Like, here, let me bring it up real quick. I want to see what it said again.
There's just all this bullshit around the terms that we use for things. Analogies and all of that. And the pseudoscientists have latched onto it, and they're just beating us over the head with it. And I'm not. I don't know. Should we even pay attention to people like Standing?
I don't know. Well, it's. I feel like you said once, like if we let them get off the rails, then soon pretty much everyone's going to be off the rails.
Yeah, people listen to him because he sounds so confident. You know.
Yeah. All right. Here's what it says. It has long been known that some non-coding DNA has important functions. This was known even before the phrase junk DNA was coined.
Right.
However, there is good evidence that much DNA has no function. So it was known, according to Talk Origins, that a lot of DNA had a function even before the term junk DNA was created.
Yeah. They knew that there was non-coding functionality. I mean, it was talked about back in the 60s and 70s, and I don't know.
Yeah.
And I think the thing is, junk DNA has been popularized by the creationists, not science at all. Yeah. And they make it sound like it's absolutely essential for evolution. And it's just, it is just such a ridiculous thing. So, Knock, I wanted to talk to you about this addiction you have and how you've... You know what I'm talking about, right?
Weed?
No, not that one.
All right. So real quick, I've got to mute. My daughter's calling me, so I'll be right back.
Go ahead. Anyway, But yeah, we got to get into a little bit of that because there's so much confusion about it, and they keep. And the thing is, encode, like the whole purpose of encode was to find out all of these very, very tiny active, like 1 or 2 molecules that end up being transcribed. And it was surprising how much gets transcribed. And that is powerful evidence for, or not evidence, that's powerful machinery for the neutral theory of evolution.
Right.
Basically, this transcription is very easy because transcription happens according to consensus sequences. So if something is like if you had duct tape, which is really sticky, and you had some Scotch tape, which isn't as sticky, and you had some syrup, and you had some orange juice, I mean, it would all stick at a different rate, right? But a little bit is going to stick to almost anything. I mean, wet your finger and you can pick up dust with it, right? So the consensus, and I'm using fucking analogies, the consensus sequence basically fits to a lesser or greater degree. And it turns out that a lot of this stuff gets transcribed in the nucleus. And that does not mean that it's highly functional. Like SFT likes to say, highly functional.
Right.
Weird. It's just, I don't know.
Yeah.
I want to say that SFT is morally decrepit.
Is he?
I'm fairly certain he's an IFB. I'm almost positive of it. I'm almost positive.
I sometimes wonder where he's coming from because every now and then
I see him all over the NIFB sites. So, and it would fit the young Earth creationist mold of the NIFB. And
really, he's on those sites.
Oh yeah, all the time. All the time.
Well, that's interesting. The other thing is the people he hangs out with, like this Jason Ezard. Until Jason went after him, Jason Ezard is a real piece of shit person.
I've heard that name before, and I'm not placing it, but I've definitely heard the name.
Faithful, Honest, and True. That one.
Oh.
He was on crazy.
Yeah, all right.
Yeah. And then he got mad at SFT and he started doxing them. So
is that where that came from?
Yeah.
I'm not going to say it like publicly because I'm not a doxer, but I was wondering, like, how did people find that shit out?
Yeah. Well,
wait, hold on. I need confirmation of something. Somebody doxed,
SFT was definitely doxed without a doubt. Yeah.
Ooh, I was wondered. Never mind. We'll talk off air.
Yeah. Yep. But anyway, we were just wrapping up. So, what you doing anything tonight?
Who, me? No. And it's really weird. Your StreamYard does not like my scripts. So when I say hack, I mean scripts. Your StreamYard does not like my scripts. Like everything's messed up. Like I restarted my computer and it's still all jacked up. I'm like, I'm not sure what that's about. But my scripts don't like your StreamYard. But it's fine. Whoever the person, I don't see a name because of my scripts. But there's one person that I don't see in StreamYard. I don't know who you are. You are smarter than I am, though. I'm just going to give you props.
Rational empiricist? You mean Andrew?
Yeah, I don't see him in my StreamYard thing. It's really, really weird. Your StreamYard does not like my scripts at all. I can't speak.
Scripts.
Speed, I've become a ghost.
Well, like, so I've got this like program. I got it from CC, Critical Cripple, and it allows me to use OBS with my StreamYard.
Yeah.
And there's scripts inside of StreamYard that allows me to do it. And your StreamYard does not like it at all.
Top Dog and I set up a couple of streams today using Zoom, coming into OBS, Streamlabs OBS or Slobs or whatever, and on my Mac, and it actually worked. And we got the sound out there, so I don't know. I'm thinking about switching over to using Zoom, getting rid of StreamYard entirely.
Well, StreamYard has definitely had issues lately. I don't know what their deal is. They need to fix some shit. But I know like on our channel, we just pay, like John paid for a whole year of StreamYard, and now like StreamYard's completely fucked now.
Yeah, I'm paying monthly for like $40 a month, and they're pissing me off more and more. So if I can go to Zoom and Zoom works up, so we're gonna try a few streams with Zoom and
In Zoom,
Yeah, well,
Zoom also
yeah.
had issues because of the back door. I don't know if you guys heard about that, but they were
What's that?
hacked. Zoom also, there was this whole.
It's
big
a China
scandal.
company.
Yeah.
Well, what are they doing with?
I don't know, but the information's out there.
Yeah, and so I stopped using Zoom because there became an issue, like for and this happens with streaming shit. So like Zoom, for some reason, for a period of time, you could not use it on OBS. You could not do it, and then they fixed it. But by the time they fixed it, I had already went to StreamYard.
Yeah, well, that's a
Yeah.
problem because
Yeah.
they could shut me down. But I could always go back to StreamYard free and have a
Yeah. And it is supposedly fixed now, so you should be able to use Zoom with OBS now. But for about 2 months, you couldn't, and that blew up my whole shit. I started using StreamYard.
Yeah. That's what modern day debate uses, right? Is Zoom. And,
Yeah, they use a combination. I think they use Restream, Zoom, and sometimes StreamYard, depending on who's moderating. And I think James is basically like Restream, Zoom, and OBS.
Oh, and it praises moderating. He's StreamYard.
I think he's StreamYard. Yeah.
Yep.
Yeah.
Okay, I think we'll end the broadcast and go backstage. How about that?
Yep. Yeah.
Oh yeah. I was just jumping in because like I told you I'd jump in earlier, so I'm like I'm here now.
Stick around for a minute. Going to end the broadcast. Goodbye out there. Thank you all for
Goodbye.
stopping.